Diet and nutrition contribute to both beneficial and harmful aspects of oxidative processes. The harmful processes, termed oxidative stress, occur in nearly every human disease, especially those that are cardiometabolic in nature. Integrative and functional nutrition includes a focus on individual, family, population, and environmental aspects of health, which is inherently holistic in its approach. Despite this, knowledge attained in the field of nutrition science has largely relied on a reductive logic. This assumes that by understanding the component parts of a system, we can understand the whole. This approach has served us relatively well in understanding essential and non-essential nutrient functions and how genetic variations require targeted nutritional interventions. A wonderful example of this approach working is the management of genetic variants of phenylketonuria and the use of tetrahydrobiopterin. Reductionism has provided important details regarding oxidative stress as well. This includes the causes and mechanisms, as well as knowledge of dietary oxidants and antioxidants. With the dawn of new technologies in genomics and other omic sciences and systems biology approaches, we've now seen expansion of our ability to study how the functional outcomes on the right of this figure of an individual will interact with an array of oxidant exposures and dietary factors that you see on the left. This interaction occurs through a redox interface, which you can see at the center of the image. And in this interface, multiple systems are working together to adapt to environmental changes and resources to ultimately influence our health. Some examples of things that are helping to mediate these interactions are things like cysteine and glutathione. For oxidative stress, we've only seen modest advances in knowledge over time, and the use of omics technologies and systems biology approaches will be paramount for continued understanding of nutritional impacts on oxidative stress. So you've probably heard about oxidative stress in one or more of your classes so far at ASU. Oxidative stress is really a two-sided thing though. On one side, too much oxidation or oxidant exposure can cause damage to biomolecules. The definition of oxidative stress has evolved over time, and it's currently defined as an imbalance between oxidants and antioxidants in favor of the oxidants. This leads to a disruption of redox signaling and control of different molecular processes and can ultimately result in molecular damage. On the other side of the coin, maintenance of a physiological level of oxidant challenges, or oxidative eustress, is really essential for managing processes and metabolism through redox signaling. Redox signaling is the transmission of a redox signal through an essential element of redox from a source to a target. So basically moving an electron from one compound to another to manage a signaling cascade or to get rid of a free radical. Free radicals have one electron, but there are also non-radicals that contain two electrons, and believe it or not, both can facilitate the transfer of reactions in biological systems through electron transfer. Disruptions in either free radical or non-radical electrons can result in oxidative stress. Free radicals are reactive and unstable molecules that lead to chain reactions of oxidation. Targets of free radicals, like hydroxyl radical, are things like vitamins A and E, which you've probably seen covered in some of your nutrition courses. Although free radical reactions are a common target in nutrition and oxidative stress research, data show that scavenging mechanisms in biological systems limit the propagation under most conditions. This makes non-radical reactions more common under most oxidative stress conditions, which is sort of the opposite of how it's often presented in early nutrition courses. So what targets these non-radical oxidants, things that don't have an unpaired electron, like oxygen, which can become superoxide, and hydrogen peroxide? Well, there are these things called master switch regulators, which utilize as cofactors minerals like selenium. And we're going to come back to that topic in a minute after we do some background exposure to this context of master switch regulators. But first, let's talk a little bit about the broader scope of this. Across a lifespan, individuals are exposed regularly to dietary oxidants, glycooxidative stress, so this is a connection towards aging, so there are these things called advanced glycation end products that occur through the aging process. There's endoplasmic reticulum stress. The endoplasmic reticulum does a lot of work inside of our cells. Our proteins do a lot of work, so there can be stress applied to those proteins. We can have disulfide stress. Disulfide bonds are a way of regulating different metabolic processes, and if that shift between singular sulfides and disulfides occurs, we can have some problems. We're exposed to UV stress every day through UVB rays. There's ionizing radiation in the environment that we're exposed to. Nitrogen-containing compounds in our body can also undergo stress. And then, of course, there are nanoparticles, air pollution, and a variety of other environmental toxins that there's really not much we can do about because they're everywhere around us. So how do we combat these complex stressor systems? Well, we have many antioxidants around us. This includes vitamins like vitamin E and vitamin C, so the tocopherols, the tocotrienols, and ascorbate, as well as other direct antioxidants like glutathione or ubiquinols. It also includes other nutrients though, like selenium, iron, copper, zinc, riboflavin, and other B vitamins. These are all needed to support antioxidant systems in our body. Many of these things are like a hot potato reaction of passing electrons from one molecule to another to regenerate the final reactive electron quencher. The spectrum additionally includes thousands of dietary components that affect oxidative metabolism and oxidative stress, such as carotenoids, which are plant-based forms of precursors to vitamin A. There are flavonoids, organosulfur compounds, phenolic acids, polyphenols, and things like tofu, still beans, and tannins, which are in a lot of teas and coffee and other things that have brown components to them. So thinking about this as a more complex system, how do we achieve redox balance? Redox balance is maintained by preventing, intercepting, and repairing free radicals, which together are also important for generating regulatory potential for molecular thiol-driven master switches. So an example of some master switches, which you definitely don't need to memorize, but may be of interest if you want to read more, are NRF2 and KEAP1 and NF-kappa B. These molecular switches are used for system-wide oxidative stress responses. Mechanisms for redox. There are many theories that try to explain how redox reactions occur. So These provide tight control of plasma thiol or disulfide redox potentials despite diverse environmental exposures and oxidative stress. And when we understand the intricate balance between antioxidants and oxidants, we can start to understand how diet plays a role in oxidative stress and disease outcomes. On a previous slide, we saw that non-radical species like hydrogen peroxide or singlet molecular oxygen, rather than free radical species, perform the majority of secondary messengering functions. Chemokine-controlled NADPH oxidases, which are utilizing niacin as a cofactor, and metabolically controlled mitochondrial sources of hydrogen peroxide, as well as glutathione and thyroredoxin-related pathways, have powerful enzymatic backup systems, and these are responsible for fine-tuning physiological redox signals. So in redox theory, NADPH plays a central role in supporting redox regulation and defense against environmental oxidants. And you can see that sort of intricate connection in the figure on the right of this slide. So we've got NADPH interacting with mitochondrial respiration and NADPH-dependent oxidase enzymes at the bottom, and that's driving oxidation. But at the same time, NADPH at the top is interacting with other enzymes to create a steady state to combat that oxidation through reductive pathways. And these create this intricate balance which ultimately controls glycolysis, how our cytoskeleton functions, how transcription of gene base pairs occur, how we translate RNA into a protein, and even how cells undergo apoptosis. So redox reactions are much broader than many of us have really been exposed to in prior coursework. And I want you to just have an appreciation for that. And this occurs through kinetic control of oxidation reduction potentials, and also how cysteine residues on proteins are regulated. So again, let's take a step back and look at a larger picture for redox control using the nutrient selenium. The exposome, which you can see on the left side of this figure, represents a composite of every exposure to which an individual is subjected from the moment that they're conceived until their death. This includes environmental exposures, behaviors, as well as the interactions between those exposures and behaviors in that person's own genetic composition. Selenium is an essential mineral cofactor in redox reactions. Selenium-dependent enzymes will help to maintain molecular structure and function, and also participate in both physical and chemical barriers between an organism and its environment. This leads to the redox interface, which you can see at the middle of this picture. This interface is a network of systems that function together to enable beneficial effects and protect against harmful environmental exposures. This leads us to the left side of the screen. Here, selenium has a relatively limited number of metabolites and is required for only a small number of proteins. However, from the small network mapping that you see between the middle and right side of the image, you can see that many functions are influenced by selenium in the diet. These include various omics and genetic spaces, including our genetic composition, the epigenome, which is how we regulate which genes are expressed in our cells, the transcriptome, which is the RNA that are expressed, the proteome, which are the proteins that are expressed, and also the metabolites that are produced by those proteins. And selenium is involved in all of them And this comes back to those molecular switches. There are a few switches that selenium helps to turn on and off to regulate redox balance in cells, which drives metabolism, which drives physiology, and ultimately influences our risk for health and disease. So let's go back to that redox theory image from earlier and plug in selenium where it supports these redox systems by regulating the proteome, or proteins involved in key reactions. The redox proteome includes reversible sulfur switches, which serve to coordinate and regulate functional systems. This system operates with kinetic control through steady state oxidation of proteins that have a cysteine residue. The oxidation occurs because of hydrogen peroxide that is allowed to be present in the environment. So typically we think of hydrogen peroxide as bad, but in small regulated amounts, it's critical for being able to turn these cysteine switches on and off on proteins. It's not always hydrogen peroxide that does this. It could be other oxidants, but all of this then is counterbalanced by that niacin-driven steady-state reduction, and this occurs through thioredoxins, which is represented by the TRX, and also glutathione, which is represented by the GSH at the top center of the image. The hydrogen peroxide availability is kinetically controlled by selenium-dependent peroxidases and other control systems, and then again, the thioredoxin system is also maintained by selenium-dependent thioredoxin reductases, or TRXR. So ultimately, you can see that selenium is involved in these master switches, which is also utilizing niacin. So there are these multi-nutrient interactions occurring to regulate these complex systems that span from the genome all the way to the metabolites that are produced from protein reactions. So what takeaways can you have from this short mini lecture? I think the main takeaways are that redox reactions are critical for maintaining molecular and metabolic activities involved in health and disease. I hope that you walk away knowing that oxidation is not always a bad thing, and our bodies actually try to balance to promote a little tiny bit of oxidation in all of our cells and organs to drive these important redox-mediated molecular reactions that allow us to metabolize energy appropriately and manage our inflammation and immune systems effectively. It's not until there's an imbalance in these redox activities that favor oxidants at a large amount that we result in oxidative stress and disease. So when treating a patient, those are things that you need to be thinking about. Oxidative stress is important in the etiology of nearly every cardiometabolic disease, including cancers. And in order to adequately treat oxidative stress, that requires a holistic approach to understand where that person's at, what their personal story is, what environmental exposures they've had, what their genetic composition is, and bringing all of those pieces together to really understand the culmination of factors that influence redox activity and ultimately health outcomes.